When a cell divides, each daughter cell must receive its full complement of genetic material in the form of chromosomes containing DNA or deoxyribonucleic acid. A chromosome is made up of two long strands of DNA and several types of proteins. A DNA strand is composed of a long backbone of sugar and phosphate units, one of our different nucleotide bases A, T, C, or G, hand off each sugar unit. The sequence of the bases encodes genetic information. The three steps in the process of DNA replication are initiation, aggregation, and termination. Replication basis. Replication depends on the pairing of bases between the two strands of DNA. The A base can only bind to T, and the C can only bind to G. In a DNA double helix, the bases of one strand face across the bind and to the other strand. Therefore, the base sequence of each strand complements that of the other. The sequences are antiparallel and serve as templates of each other's replication. The two strands are labeled by the location of certain chemical bonds in the DNA backbone. A cell can replace the leading strand as a single unit, but must replicate the lagging strand in small pieces. Initiation. Replication begins at a location on the double helix known as OC, to which certain initiator proteins bind and trigger unwinding. Enzymes known as helicases unwind the double helix by breaking the hydrogen bonds between complementary base pairs, while other proteins keep the single strands from rejoining. The proteins surround the unzipping strand and relax any twisting that might damage the unwinding DNA. The cell prepares for the next step, aggregation, by creating short sequences of RNA called primers that provide a starting point of aggregation. Aggregation. With the primer as the starting point for the leading strand, the new DNA strand grows one base at a time. The existing strand is a template for the new strand. For example, if the next base on the existing strand is an A, the new strand receives a T. The enzyme DNA polymerase controls aggregation, which can occur only in the leading direction. The lagging strand unwinds a small sections that DNA polymerase replicates in the leading direction. The resulting small Okazaki fragments can contain 1,000 to 2,000 bases in bacteria. But eukaryotes, organisms have cells with nuclei, have fragments of only 100 to 200 bases. The fragments terminate in an RNA primer that is subsequently removed so that enzymes can stitch the fragments into an elongating strand. Termination. After aggregation is complete, two new double helix have replaced the original helix. During termination, the last primer sequence must be removed from the end of the lagging strand. This the last portion of the lagging strand is the telomere section, containing the repeating non-coding sequences of bases. Enzymes sniff off a telomere at the end of each replication, leading to the shorter strand after each cycle. Finally, enzymes called nuclease proofread the new double helix structures and remove mispaired bases. DNA polymerase then fill in the gaps created by the exercise bases. So in this section, as the polymerase goes through, break the whole DNA helix, double helix. So it breaks through the polymerase, and then the leading strand is top 5, bottom 3, the DNA polymerase. So and then the helicase single strand binding proteins and Okazaki fragments build a leading strand, build a new DNA double helix over there, and then it's the same thing over there. So one single DNA double helix makes two single double helix. So that is called DNA replication. Thank you.